Hello there and welcome to this Biology Mind A-Level tutorial on studying cells, in which we'll be having a look at electron microscopes. So in our last lesson we learned about the features of light microscopes and how they work. We looked at how light microscopes use white light to illuminate a specimen which we can then see. You now understand that stains are used to enhance the image of the specimens and how dry mount and wet mount slides can be prepared for viewing with a light microscope. So we have four key learning objectives for this tutorial. Number one, to have a look at the transmission electron microscope. Number two, the scanning electron microscope. Number three, staining in electron microscopy. And number four, the benefits of light over electron microscopy. So let's kick off. So in our last tutorial, we talked about how there are three main types of microscopes. Number one, optical light microscopes, which we talked about in the last tutorial. Number two, electron microscopes, which we'll talk about this tutorial. And number three, confocal microscopes. So let's talk about electron microscopy. So they use, as the name suggests, they use electrons to form images. Now, electrons are smaller than light photons, hence they have a much greater resolution. So because they are smaller than light photons, they have a much greater resolution than optical microscopes. So the maximum resolution of an electron microscope is 0.002 micrometers. And they can be used to produce very detailed images of tiny structures. And we call those images electron micrographs. Okay, so the maximum magnification is times 500,000, so much more than our light microscope if you compare that to our last tutorial. An important note is that electron micrographs are black and white. So electrons cannot be seen by the human eye, hence they are projected onto a fluorescent screen to form a black and white image. So there are two types of electron microscope. There is a transmission electron microscope and number two, the scanning electron microscope. So a transition electron microscope produces very high resolution images. And the transition electron microscope projects an electron beam through a sample and a 2D image is formed. Now it can be used to produce very detailed images of cell organelles, as you can see in this cell here. For example, you can see the stacked grana of chloroplasts. So remember, the grana are this unit here, which is the thylakoid membrane stacked up on each other. So you can see them here, all of those stacked membranes on top of each other. That's our grana in our chloroplasts, and you can see that on electron microscopy. Now, denser tissue appears darker in the micrograph as the electrons are easily absorbed. In less dense regions of a specimen, the electrons can easily pass through, making them appear lighter on an electron micrograph. Now, TEM has some limitations. So number one, it must be performed in a vacuum. So it cannot be formed in normal air. Now, the caveat to this means that living specimens cannot survive, or they cannot be viewed under a TEM, because living specimens cannot survive in a vacuum. There's no oxygen in a vacuum, so TEM cannot be used to visualize living material. Another limitation is that it must be performed on very thin segments, because thick specimens easily absorb the electrons and therefore do not produce good images. So those are our limitations there. Now, in contrast, scanning electron microscopy directs an electron beam across a sample, and electrons are captured by a cathode ray tube, and the scanning process releases electrons from the specimen which are captured in a cathode ray tube. And SEM can produce 3D images of a specimen, whereas TEM produced 2D, remember. So SEM can scan the surfaces of a specimen and capture all the textures, and it can therefore be used on thick specimens. However, a limitation to SEM is that it has a lower resolution than TEM. However, it still provides a higher resolution than a light microscope. Now let's think about staining in electron microscopy. So unlike stains for light microscopy, which are dyes, the stains for electron microscopy are heavy metals. Now, the metal ions cause the electrons in the specimen to scatter, which causes some areas of the specimen to appear darker than others. So the most commonly used stains are uranium and lead. Now, the benefits of light over electron microscopy are as follows. So number one, light microscopy can be used to visualize living and non-living specimens. Whereas remember we talked about how our vacuum means that we can't view living specimens, whereas light microscopy can be used to visualize both living and non-living specimens. Light microscopy is relatively quick, whereas electron microscopy is a more lengthy process. And electron microscopy is much more expensive to run than light microscopy. So that is our tutorial complete for today. I shall see you for the next one.